time. And when I fit a cubic function to that, you can see that there is a significant correlation between sperm viability and temperature across that range, with a kind of local maximum right around the normal, a fairly normal hive temperature, around 30 degrees. So if you set a somewhat arbitrary tolerance threshold of 20% loss of sperm viability as being acceptable, then we can say that between around 13 degrees and 39 degrees is safe for queens. I'd be comfortable making that statement. And that outside of that range, so on either end, then things start to get a little bit too risky. But my real goal here is to try to find some protein biomarkers that can indicate if a queen has being temperature stressed. So I chose to look at heat in the beginning because, as you can see in the graph, that up here, the hotter temperatures seem to have a more dramatic effect. So I began by looking at heat, and I took 20 queens that were all originated from the same colony, and then I treated 10 of them at 42 degrees for 2 hours, and kept the other ones at a happy 30-degree temperature. And then I dissected them and extracted protein from their ovaries and spermatozoa, and measured the protein abundance in those 2 organs, comparing the hot and control treatment groups. So if you're not familiar with proteomics, all you really need to know is that this is a technique that lets us quantify thousands of different proteins all at the same time and compare abundances between 2 different treatment groups. And if you are familiar with proteomics, then you might be interested to know that this was all done by label-free quantitation using a Bruker QTOF, but that's only for a few people in the audience. So I was also interested in looking at virgin queens because I wanted to be able to say something about whether these differences in protein expression were from proteins expressed by the queen herself, or if they were proteins that might be changing in the sperm cells. So obviously a virgin has no sperm stored in her spermatozoa, so that comparison is purely going to be things that are changing in the queen. And this is a volcano plot. If you're not used to looking at that, all the dots that are beyond those sweeping lines to the right are proteins that were significantly upregulated in the heat-shocked queens. The ones on the left of the line are ones that were significantly downregulated. So what we found was that for the mated queen spermatozoa, all the proteins that were upregulated were heat-shock proteins. Maybe that's not a surprise. For the virgin queens, 2 of the exact same heat-shock proteins were also upregulated, and the other 3 actually showed the same expression pattern. They just didn't pass the significance threshold set. For the mated queen ovaries, a different heat-shock protein was upregulated. So you might look at this and think, Allie, you just told us the most boring result ever, that heat-shock proteins get upregulated with heat shock. But I would like to try to convince you that it's actually more interesting than that. So those heat-shock proteins can be divided into sort of 2 categories. And one of the categories uses ATP, so they're ATP-dependent heat-shock proteins. The other category functions independently of ATP, so they don't need it in order to work. And all 5 of those proteins that were upregulated in spermatozoa, they were all of the ATP-independent type. And the single heat-shock protein that was upregulated in ovaries, that one uses ATP. So I sort of have a hunch that this is not a coincidence, that there is actually some evidence from other work that people have done that this might be sort of a more deliberate, if you will, strategy. So Boris Barr, him and his colleagues have found that the ATP economy in a spermatozoa is really important. It's tightly linked to sperm longevity. So they created a cool system where there's like a little spermatozoa in a tube, so an in vitro system that mimics what is going on in the spermatheca, and they found that when that is supplemented with metabolites, the kinds of metabolites that allow for the best longevity are the ones that produce the most ATP per molecule. So if lots of ATP is good for sperm longevity, then what this is showing is that a mechanism of stress response that conserves ATP has turned out to be favorable. So also, you'll notice that somewhere around 20 different heat shock proteins were actually identified, and only those 5 were the ones that were actually upregulated. So from a purely practical standpoint, it's important to know, for the sake of biomarkers, that those 5 are the 5 that we should expect to change with that amount of heat for that length of time. So then what we did was, with the help of Robin Underwood, who donated these failed queens to us, is we looked at the expression patterns for those 6 different proteins in queens that actually had failed in the field. And what we found was that the queens, or 5 of the queens that failed in the field actually had very similar protein expression patterns to the queens that we had heat shocked in the lab. So what's shown in this heat map is just the expression of those proteins clustered with the lab heat shock samples. So this does not confirm that they work as biomarkers, because we don't actually know what those queens experienced in the past, but what it does illustrate is sort of how these biomarkers could work and how we might use them in a more practical sense. What we still need to do is confirm with queens that have actually been heat shocked in sort of a blind field trial to see if I can correctly categorize the queens after some length of time. That's something we're working on in the future. So in summary, what we can say based on this data is that temperatures of 13 to 39 degrees are safe for queens, and that queens upregulate specific heat shock proteins, so those 5 in the spermatheca and 1 in the ovary, in response to heat, and that some of the queens that failed in the field, granted it was only 5 out of the 11 that we looked at, but at the same time we wouldn't expect all of the queens that we randomly sampled to have been heat shocked. So some of the queens that failed in the field showed a really similar protein expression signature as those we heat shocked in the lab. So where is this going in the future? Well, what I'm working on now is expanding the stressors to look at, say, cold shock and pesticide exposure to try to find unique markers for all of those things. And then what we'll need to do is figure out the sensitivities, how much of a stress is required to see these proteins upregulating queens, and how long that signature lasts for. Those are sort of more practical things that we still need to investigate. And something I'm really excited about right now is looking for those markers in more queens that failed in the field. So once those panels are developed and confirmed, then what I want to do is take queens like these ones that we've received in a failed queen survey that I've been doing in collaboration with a lot of beekeepers in BC, and Abby, who's here in the audience. We gathered all these different queens, and for the ones that show sort of more cryptic symptoms, like the drone layers, the poor buildup queens, and the poor laying queens, I want to examine those queens to look for these different markers so that we can say this percent of the queens failed because of this stressor, this percent failed because of heat, and that type of thing. So that then once we know which stressors are the most important, then we can do something in our operations to try to avoid them or at least mitigate them in the future. So thank you to my advisors, obviously, and everybody who has helped with this project along the way, particularly Kappa for giving me a travel work to come here and share this with you.
Thank you very much. No time for questions. I'm very sorry. It's my first session I moderate where there's no discussion at all. So the next talk is in-depth proteome and hypopharyngeal glands of honey.